Okay, so after the long extended warm-up overview, we wanted to dig in the next two talks on two topics. So one is today, tumor evolution, and then tomorrow morning, Niko will do tumor progression models. So tumor evolution, remind you after the short break, right? The cancer is this evolutionary process, accumulation of mutations in a population of cells, somatic mutations. And so now we want to quantify this process. So of course, as an evolutionary process, there's selection happening. So cells that acquire a mutation that gives them a selective advantage will undergo some will expand in the population. So that you get these subsequent waves of clonal expansion. This is the clonal theory of cancer. So now, well, we'd like to quantify this. Well, why? I mean, one, because of course it's interesting to try to understand it for basic cancer biology. Basic cancer biology, understand this process. But actually, understanding this process and understanding something about the order of mutations is also being recognized and being important clinically. So this is just one example from a recent publication of comparing patients with a certain type of leukemia and looking at the order of two cancer-causing mutations. And what was sort of observed was that one order of these two mutations in these two genes, if you got this mutation in this JAK2 gene first, that led to worse prognosis for the patients than if you got the mutation in TET2 first. So there's inevitably probably more of these things to come out as we're able to quantify this progression process, this evolutionary process further.
But do you have also an example where a drug was, like drug treatment was affected by the order of mutations? This would be the interesting thing, right? To show that you can predict it and then you can tailor the treatment based on it.
Right. So in this publication, there was some hints that these patients responded differently to a drug treatment, but I don't recall the details. But yeah, that of course would be even more important clinically. So population of cells, we want to quantify this process. How do we quantify evolutionary processes? We try to write them down in terms of trees, right? So if we've measured a tumor and we've sequenced this tumor, and we've sequenced the cells in the tumor, and for now let's assume we've measured every cell, then we can describe the process of mutations accumulating in this population via a tree, here a cell tree. And so what we'd like to do then is, from the data at the present time, from the cells and their mutations, try to reconstruct a tree that describes this ancestral relationship. So for this talk, we're going to describe ways to build trees or to quantify this evolutionary process using two different types of models. One are phylogenetic tree models, and then secondly, models that deal more with the population dynamics. So there'll be these two parts. Okay, so first, basic phylogenetic trees. I mean, this is a huge topic. We're not going to give an intro, can't do justice to giving an intro to reconstructing phylogenetic trees and data in the context of this talk. But again, we measure. We have a tree. We measure the leaves of this tree. We're going to make this more precise in another slide. And we'd like to reconstruct a tree that describes these relationships. So of course, this is done in many different ways. Huge literature on how to reconstruct trees, describing the relationships between species, between all sorts of things. You need a model to do this. There's different approaches. This approach is based on distances. If I have only distances between these species, I can try to fit those distances between a tree, between trees. Of course, in the era of genome data, as was already sort of described this morning, Lior sort of alluded to the fact that now with genome data, we really can use these sort of character-based methods. So at a high level, we can think of each cell exhibiting some characters which we can measure. These could be individual positions of the genome, and we've measured whether there's an A, C, T, or G or at that position. So these could be our characters. They could be something more abstract. They could be whether or not there was a somatic mutation at the position. They could be whether there's a copy number aberration. It could be all sorts of things. But we've measured these characters for each cell. We've measured some number of characters. These characters have a certain state. So here, D states for the characters. And what we want to reconstruct then is a tree tree that describes the relationship between these cells. Again, the cells are the leaves, and in our case, to make this a little more precise, we'll be interested in finding rooted trees, because in the context of somatic evolution in cancer, we in fact know the root. The root is the germline genome, so there is always a rooted tree that we can think of. We're interested in a binary tree, because if we're modeling the process of individual cells divide into 2 daughter cells, right? Not 3 or 4 or 5. So if we had a cellular resolution and we could reconstruct everything, we'd have this rooted binary tree. Now, of course, all the difficulty comes in, what model do you use to describe how characters change over time? And then based on that model, you then have to try to find the tree that maximizes some objective function for that model, right? So we could, for example, count changes in characters and then try to find the fewest number of changes that describe the species. We could have a probabilistic model that describes the probability of a character changing to another character, and then try to find write down a likelihood for each tree, and then try to find the maximum likelihood tree. Both of these problems in any reasonable construction are challenging problems. They're NP hard problems, but there's all sorts of great algorithms and approaches that one can use. So let's formulate a simple model to make this a little more concrete for somatic mutations. We'll start by assuming all somatic mutations are single nucleotide variants. So forget about copy number aberrations and structural rearrangements and all this crazy stuff. Just assume that the only somatic mutations are single letter changes in the sequence. Secondly, we're going to make another assumption, the so-called infinite sites assumption, which is to say that a mutation occurs at a position only once during the evolutionary process. This assumption is one that, for tumors if we're thinking about single nucleotide mutations, it's a simplifying assumption, maybe somewhat reasonable. We expect that the genome's pretty long. The chance that, randomly a mutation would happen in the exact same nucleotide is pretty low. So let's assume that probability is 0. We have the so-called infinite sites assumption. So then, according to that assumption, then every character, single nucleotide variant, is our character. If a mutation only happens once at a position, it means that we can just measure that position as a binary character. Every position in the genome is either mutated or not, and so we build this binary character matrix describing our measurement, our cells. Now, I should say, and I think it was on the slide and I forgot to say it, that the other assumption that is made in character-based phylogeny is that the characters are evolving independently. All right, so we've taken independent characters for single nucleotide variants, of course, independent positions. This assumption will break down. So now, given this, it will break down for more complicated rearrangements. Given this matrix, we'd like to reconstruct the phylogenetic tree. It's a very simple model, the so-called rooted binary perfect phylogeny model. Okay, so let's describe the trees that exhibit this model. Rooted trees, the root is the normal or unmutated state. Again, this is our assumption because we're evolving from the germline genome. The leaves are the cells that we measure. Each node is labeled by this binary string indicating which mutations are present at that node. So here's the one we measured, the ones we measured at the leaves, as well as potentially some other. Since a mutation happens only once, every mutation labels only one edge on the tree. And the edge labels on the unique path to the root actually spell out the mutations that are present in that cell. Okay? Very straightforward model. Turns out, and this is so-called perfect phylogeny theorem. Here, the reference is to Dan Gusfield's work, although I guess it's also in the book, so you should buy the book and read more about perfect phylogeny. This is, in a sense, the simplest possible version. It's the rooted version, binary characters. We have the ancestor. We know the ancestor. It's the all-zero state. Okay, so then there's a nice characterization first telling us if I give you such a binary matrix, does it exhibit this rooted binary perfect phylogeny? And in fact, all you need to do is check pairs of columns and see that that no pair of columns contains all three of these states 0, 1, 1, 0, 1, 1. So-called 3 gametes condition here. Some of you may know the 4 gametes condition when you have when you don't have the all zero root. Yeah, question.
So you're assuming that the mutation rate's fast enough that you get multiple mutations along a branch? Cancer?
Multiple. We don't know. We don't need multiple mutations on a branch necessarily.
Can you define the colored dots on the edges then?
Yeah, these colored dots here are the mutations. I don't necessarily have multiple mutations on a branch. I just have, well, I mean, essentially on some branches I don't even have any mutations, so that's why I've kind of dropped them down here.
There's an edge with a red and a green dot. What does that represent?
Ah, right. So this one, yes, this is where there were sort of two mutations on this branch. That's the sort of tree we would resolve, and I guess we would resolve it this way because we really start with we measure the leaves here, and then we're trying to sort of build the tree that fits this data under the assumption where there's at most one mutation. So I could actually have put in a node here that had an edge with just the red dot, and then a node, and then an edge with just the green dot. It would be this chain, but I could delete that node because it doesn't need to be there. Good question. Simplest possible model, you can reconstruct the tree in time linear in the matrix, right? So great, everything works. The model, of course, is very simple, but yeah.
Is it a unique solution?
It's unique. It's unique under. Let's see if I'll get this right, and then Dan can correct me. If the columns, if there are no repeated columns, I think you'll get uniqueness. Yeah, in this simple one, yeah.
Binary tree, right?
Oh, this one. Well, in my cartoon, this one's not binary, unfortunately. I said I wanted a binary.
The assumption is for binary.
Sorry, binary is not binary tree here. It's binary. It's binary character. It's 2-state character. Probably would have been better if I said 2-state character, not binary tree. 0, 1 characters. That's what. Yeah. Okay, so I mean that was really simple. So. What happens in tumor phylogeny? What are the complications? Why is there anything to do that's different from all the other work that's been done in phylogeny for the past several decades? Yeah.
One more question. So if there is that one edge at the very top, which all of them share that mutation, those 2 mutations.
Yeah.
How can you tell that those are mutations?
Right. Well, we'd see these mutations. Remember, we're detecting somatic mutations. So we'll assume that this process that we discussed in the previous lecture of detecting somatic mutations has worked. And the way we knew they were there is we compared to the germline genome. These are interesting mutations. These are mutations that one could think are, the early ones, before some of these later clonal expansions, right? Not all of these are interesting. In fact, a bunch of these mutations are in every single tumor cell. They're probably just a bunch of them are presumably passenger mutations that accumulated in that first founder cell of the tumor before it even became cancerous, right? So any random mutation in that, cellular lineage would be there as well. So there tend to actually be a fair number of mutations up here. In some sense, they're the easiest to detect because they're in every cell in the tumor. It's the ones down here that are hard. Okay, so what makes the problem harder in cancer? Some due to challenges in the measurement. So we generally for a lot of the data, as I'll show you, we don't measure single cells. We measure mixture of cells. When we do measure single cells, or even sometimes when we measure mixture, we have observation errors. We have measurement errors, and we need to deal with those. A lot of the phylogeny models that have been around don't really deal with measurement error. And then we have these issues with these complex aberrations. And so if we want to understand how those progressed in our population, then we start to run into problems with some of the assumptions that are used. So let me talk about the first challenge, and then we'll go through the other two. So this question of sequencing mixtures. Okay, so again, a lot of the data, if we don't do single cell sequencing, if we in fact just sequence this whole tumor sample, now we don't have every single cell measured. We've got them all mixed together. So now in terms of this tree, I haven't measured the leaves of the tree individually. Instead, I've given you all the leaves simultaneously. And now, can you do anything there? It seems actually hard to do anything. So what's also been done recently, especially as sequencing costs have gotten lower and as you need less material to sequence, you could chop up your tumor into multiple pieces, sequence multiple samples from the same tumor, and then try to reconstruct a tree. Some of the published trees you'll see from experiments like this, actually what they're doing is reconstructing a tree, treating each of these mixtures as actually the same thing. So if we think about it in terms of this, say, binary matrix, where we originally had a matrix on cells and mutations, really what these are, each sample is a mixture of these, so somehow this is a collapsed matrix. And so we can look at this collapsed matrix and try to use existing phylogenetic trees techniques to relate samples, but then we're not addressing the fact that somehow each of these was a mixture. We're losing information. Okay, so we presumably would want to try to recover this information. Yeah.
Matrix, can you explain how these mixtures are derived? So how do we observe 1, 1, 1, 1, 0? The
Yes.
X, how do we get the X, for example?
Yes. Okay. So let's say that this one sample had really two different cell types, right? And what we're measuring are the superposition of those two cells. So it's just the OR. It's just the OR of the rows, right? Assuming it's just the OR of the rows. So it's the superposition. Okay. So we could, of course, take this data, which is lost information, and you plug it into an existing technique. But could we do more? And this is, I think, where cancer evolution, tumor evolution, starts to differ a little bit from some of the techniques that have been used. So we could start to try to recover some of this information. For example, if we take that same perfect phylogeny model, and assume that we only looked at single nucleotide mutations, use the perfect phylogeny assumption, we could try to say, could we resolve a perfect phylogeny from the superposition of these rows? And there's been work done in this area. I'm not going to go into the details, but it's sort of a combinatorial model that tries to find somehow the minimal perfect phylogeny matrix that explains this data as superpositions of the rows.
Is there an algorithmic solution to that?
There's a heuristic. It turns out to be an NP-hard problem, as was claimed in one publication, and the proof was in a subsequent publication correctly. But there's an ILP. You can do it with ILP, yeah. But even doing that, we're losing information, because if you recall in the previous lecture, when we were talking about detecting single nucleotide mutations, we had these reads that were telling us whether or not the mutation was there. But more importantly, we can look at the reads that are telling us whether the mutation is there and actually count up how many reads have the somatic mutation. And this is the so-called varying allele frequency. So if I look at the fraction of reads covering a position that contain the variant allele, meaning the somatic mutation, then this varying allele frequency should be proportional to the fraction of cells that had that mutation. Again, assuming no copy number aberrations, right? So if every cell in the sample had the mutation, and it was a homozygous mutation even, then the variant allele frequency is 1, right? The variant allele frequency is 5, variant allele frequency is 0.5, 0.3, 0.2, 0.1. That's giving us some information about the fraction of cells with the mutation. So now we can think that our data is not mutation or not in each sample. It's these frequencies. So we could attempt to use these frequencies to reconstruct our tree. Now, of course, there's it's going to be loss of information in going from cells to what we can measure. So we cannot think of maybe constructing a tree on individual cells, but maybe at best constructing a tree on what we might call clones, which are groups of cells that have all undergone the same sort of clonal expansion. So we can only really detect mutations that are in some modest frequency of cells. So really we're going to build these sort of clone trees that describe the relationship between clones. Yeah.
This might have been on the previous slide. So are you still assuming the infinite sites model such that you only see 2 alleles at any position, or?
Well, that'll be on the next slide. So here I haven't sort of set up the model, but yes, we will be making that assumption. Here's just the data. We've measured these frequencies, right? So we want to build these trees. So now, yes, here's the problem. Given frequencies, can I find this clonal tree, right? And now you can ask the question, okay, well, what phylogenetic model do you want to use? This is actually sort of a general problem. If I take a tree, phylogenetic tree, if I give you the leaves of that tree, I can ask you to recover that tree. If I instead give you mixtures of the leaves of that tree, I can ask you to recover that tree. And now you could say, can I solve that problem under the infinite sites model? Can I solve it under a maximum parsimony model? Could I solve it under a maximum likelihood model? It's now a different question, sort of a phylogenetic mixture question, if you will. So it seems actually pretty hard. In fact, if I give you one measurement, one mixture, you really can't kind of reconstruct the tree seemingly at all. Anything could happen unless you impose some constraint. So let's impose this constraint, the strongest constraint we could impose, the infinite sites assumption. Now actually things do start to happen, because if a mutation happens only once at a position, then we actually have a relationship between the frequencies and the ancestry somehow, a simple relationship, which is that if one mutation is ancestral to another in the tree, which we don't know, then the frequency of that mutation in the sample has to be larger than the frequency of the descendant. Why? Because every cell that gets this orange mutation had to have the, wait, let me say this the other way. Right, yeah, sorry. If the gray mutation here is ancestral, every cell with the orange mutation has the gray mutation. Right? So the frequency in the cellular population of the gray mutation is larger than the orange one. This relates the two things. So now we have a constraint. With one sample, you still can't do much, because, in fact, I could take this frequency data and explain it as a linear tree just by sorting the frequencies in order. And that would always explain what happens. So there's not really much interesting to do without making another assumption. But now, what about what happened? What about the case where we measure not one mixture, but now multiple mixtures? Okay, so now I have sequenced multiple samples from my tumor. So I've taken this phylogenetic tree and I've given you multiple mixtures of the leaves in certain unknown proportions. So could you, from this frequency data, reconstruct the tree and reconstruct these mixing frequencies? And note again, this is not the tree relating the samples. We're trying to do more here. So let me sort of set up one problem. Here's our frequency matrix. Here's the tree that we generated this matrix. Could we recover this tree from the matrix? If we make this perfect phylogeny or infinite sites assumption, then we know that there's this one-to-one correspondence between trees and this special class of binary matrices, right? The ones that meet these conditions, right? The 3 gametes condition, for example, if it's the all-rooted case. So now we have these matrices. And now for any tree, it's described by one of these matrices, we're going to mix the leaves of the tree. And we mix those using some matrix that tells you these proportions in which you mix. So every sample is a mixture of the leaves or clones. Here, then, the entries in this matrix have to sum to less than 1. I've sort of I've made it less than 1 because there's also the normal cell, the one with no somatic mutations, the root. So that's the missing piece here. So this first sample is actually the previous slide here. This was a 0.8. This was just one leaf. So we had 80% of our samples. This one, 20% was the normal cells with no mutations. That's the rest of this. So that's the other property. And then how do we describe the matrix? Well, it's just a simple. It's just linear algebra, right? The frequencies are obtained by multiplying U by B, and then there's this 1/2 because under infinite sites, all mutations are heterozygous. So now, all we need to do is invert this problem. Given a frequency matrix, can you factorize it as a matrix like this U and a matrix B, a perfect phylogeny matrix? Note that both of these matrices have structure, right? So it's a special factorization problem where you have these structured matrices. I'm not going to describe the algorithms. There have been many different ways to sort of attack this problem. Some are heuristics. Some solve the problem exactly. Some solve it using quadratic programming techniques. Some do the sort of one sample case and impose extra constraints. But the other thing I wanted to say is the setup that I gave you has no errors in the data. Right? There were no errors. And it didn't deal with the fact that we need to kind of cluster the mutations, right? So as we discussed with detecting mutations and building clones, we need to deal with clustering the mutations. So there are approaches that use Dirichlet process models, but even further sort of tree-structured Dirichlet process models to simultaneously cluster variant allele frequencies and build the tree according to these constraints. You'll hear about these models further in the workshop and extensions of these models to dealing with other aberrations. That's it for me.
Okay, so I will then look at the aspect of noisy data. And if we go back to our outline here, so Ben has focused very much on the mixture aspect, trying to deconvolve the different clones that are present in one such sample. And I will focus now on the fact that the observations are noisy, and that is the case in particular for single cell data. So in a way, looking at single cells is easier than looking at the mixtures, but on the other hand, I will make this aspect of noisy observations very explicit. The other reason I will focus on single cells now is that we can do single cell sequencing now, and there's more of these data accumulating. And I think in the near future, there will be a lot of these single cell sequencing data be available. So this is just the same situation we have looked at before. Let's say starting from this ideal situation, the clonal origin of the tumor is on the top and evolution now runs from the top to the bottom. So what we would observe at the current time point is down here. There's a mixture of different clones coexisting in the tumor, and we would draw a random sample from the tumor in the form of a collection of individual cells. And each of these cells has a certain set of mutations indicated by these symbols inside the cell here. So the data, again, the perfect data that might come from such an experiment is again a binary matrix where the rows are here labeled with the mutations and the columns with the cells. And the 1 here simply indicates that this particular mutation was present in this particular cell. And a 0 would mean that this mutation does not occur in this particular cell. So you're just recording, you're just detecting for each cell the mutations that are present or not in the cell. So then what we would like to do again is from these data to reconstruct a tree. And a phylogenetic tree again would look like this. The cells would label the leaves of the tree, and the mutations would occur along the edges as you trace down a path from the root of the tree, the unique path from the root to any of the vertices of the cells in the tree. So what we think in a sense is more convenient to do here is rather than looking at a phylogenetic tree, looking at a mutation tree. So in the mutation tree, the vertices, the interior vertices are labeled with the mutations. Also there's one vertex for each mutation. And the samples or the cells, they are attached to this mutation tree. So for example, these 3 samples here are attached to this mutation. This sample has been attached to this mutation, and so on. And it's not very hard to see that, of course, under the infinite sites assumption that we continue to make here, these 2 representations are equivalent. Right? It's just a different way, essentially, of representing the situation. However, this one here turns out to be a little more convenient when it comes to noisy observations. So let's look at noise. So here's another very simple tree, a smaller one, with a root vertex here, mutation 1, mutation 2, mutation 3, only 3 mutations. And the 7 or so samples are attached to the different leaves in this particular manner. So the expected or the perfect data that you would get from a sequencing experiment when this is the underlying truth would look like this. So here are the different samples, and these are their mutation profiles. So for cell 1 or sample 1, you would have mutation 1, but not 2, not 3, and so on. And in reality, the data that you might obtain will have errors. So instead of having a 1 here, for example, you might have a 0. So you were not able to detect that mutation. You didn't call that mutation. So that will be a false negative, and there can also be false positives, like here. Rather than a 0, you actually have a 1 in your data set, so you called the mutation, but it is not really a mutation, so false positive. So this is something that standard phylogeny methods, again, do not take into account. They always assume the data is correct and perfect. And as I said in the beginning, for single cell data, it's very important to account for these false positives and false negatives because they are very frequent. So I will not go into great detail of the likelihood, but the first step here is to simply write down what the probability of this data matrix is, given, of course, the tree, and then given two more sets of parameters. Theta just summarizes the false positive and the false negative rate, and I will talk about sigma in a moment. And then this model really just says it's independent over the observations and it's independent over the mutations. It factorizes over both of these. And this probability, I'm not writing it down, but this probability is just essentially binomial. It's the probability of flipping from a 0 to a 1 or from a 1 to a 0 with these false positive and false negative rates. So that's as simple as it gets. The way we look at this model now is that we do not really care about the attachment of the samples so much. So in a way, the focus shifts from trying to estimate the dependencies among the samples to estimating the order among the mutations. Because in a way, I think this is what we would care about more. How did the mutations occur, rather than getting exactly right the evolutionary relationship between all the single cells. In a way, this is mainly what we would care about. And here are two identical mutation trees, but with different attachments of the samples. And we wouldn't care so much whether we have the situation on the left or on the right-hand side, but we do care whether we get the order and the dependencies of the mutations correct. Is there a question?
Yes.
Sorry, yeah.
So if you're dealing with single cells, how are you handling the issue of missing data in terms of dropout or insufficient sequencing?
I will not go into great detail, but the short answer is that this is a full probabilistic model. So this is hidden data in the probabilistic model. And we treat it as hidden data, so you have to do some sort of inference, either trying to estimate the hidden data or integrating it out, something like this. In fact, the example I will show you in a minute has a lot of missing data, and I hope to convince you that we get something reasonable out of it. Good. Let me try to do that. Okay, so again, the focus is on the order of the mutations, and the attachment of the samples becomes a nuisance parameter, so you would like to get rid of it, and the most natural way to do that is to integrate it out, right? And if you try to do that, it looks a bit difficult. So sigma is the vector that tells you for each sample where to attach it, to which mutation it should be attached. So sigma j is where to attach to which mutation to attach sample number j. So if you have to sum now over all these vectors sigma, that would be hopeless to ever compute the likelihood, right? Already for moderate numbers of mutations and samples, that would be hopeless. But it turns out the likelihood will factor over the individual mutation, so that's just a product here over the samples, and then there is a sum only over the possible values for each sigma j. So you don't need to consider this exponential number of possibilities, but only in this product over the samples. So in a way, the whole focus now shifts from the samples to the mutations. So you can compute the likelihood efficiently, and then depending on what you do about the prior distribution here, you can also compute the posterior probability. and that gives you a way of pursuing a Bayesian approach to sample from the posterior probability. In this way, we essentially reduced the complexity. So the tree size essentially now is the number of mutations rather than the number of samples. Now, depending, of course, on your application, that can be an advantage or a disadvantage. I think in the probably not so long a time, we will have a lot of cells, and we will probably be interested in the dependencies of a smaller number of mutations. So I think it will at some point be an advantage that the complexity is essentially driven by the number of mutations rather than by the number of samples. Although in these very first and early studies that we have right now, we don't have so many single cells because it's still costly and so on, I think that will change quickly. One more thing I would like to point out is, of course, that you still have to find the optimal tree topology, and there's no simple way of doing that. And one thing that we're doing here in this Bayesian approach is just a Markov chain Monte Carlo approach that samples from this tree space. So here's this one example that I would like to show you. It's a rather old single-cell sequencing data set. It's a very recent technology. That was one of the first papers published 2012. Single-cell sequencing data of a myeloproliferative neoplasm. And the authors there sequenced 58 cancer cells, 58 cells from that tumor. And they somehow selected 18 mutations here. That has been done by the authors. I'm not going into this. And this data set had, in fact, this amazing fraction of 45% missing data. So the technology has gotten much better. The data that's generated today has much less missing data. Still has quite a fraction of missing data. In addition, the false positive rate for this data set was estimated also by the authors, and I'm not going into it, to be moderate, right? 6 times 10 to the minus 5. Whereas the false negative rate was again very high, was 0.43 for this particular data set. So again, you cannot treat these data with a standard phylogeny approach where you just assume the data is complete and correct. We are far from that situation. This is the data matrix. Again, the samples are the columns and the mutations are the rows. And all the brown spots here are the missing data. So there's really a large fraction of missing data. Yes?
Just a comment for perfect phylogeny. So in fact, perfect phylogeny can easily handle missing data.
Mm-hmm.
So in the same time bound.
Okay.
And since the alpha is so small, and maybe the only problem is the beta, and maybe it's worthwhile to compare whatever you do using, Markov chain Monte Carlo to using perfect phylogeny. Maybe it will work.
Absolutely. Fine. That's a good point. I mean, we didn't compare it at this point. I mean, one other aspect is, of course, if you do a probabilistic treatment, you have a notion of uncertainty. We have the whole posterior, but that's another aspect. Yeah, that's a good point. Okay, so then doing this, I will nevertheless show you only one optimal tree that we found, and it looks indeed like this. So the vertices are labeled with the genes. So the mutation occurred in each of these genes, and as you see, the structure of the tree is fairly linear. It's almost a single linear path, except for this one branching point down here. So this model is completely consistent with this clonal evolutionary theory, where these mutations accumulate in individual selective sweeps, each of which is driven by one such mutation, or maybe by a subset of those. We can also look at the optimal attachment of the samples. The samples are the gray nodes here, and I'm connecting it to several vertices here when the optimal assignment was not unique, right? So, for example, this sample 19 here, all the optimal assignments are to all of these mutations on the top here, and so on. And as you see, for most of the cells, the optimal assignment is not unique, and this reflects, of course, the uncertainty that's due to this large amount of missing data and the high rates, especially of false negatives. You can also see that most of the cells are attached to the bottom of the tree here, as you would expect probably, because all of these are mutations that have occurred in the past. and most of the cells that we're observing are down here. They do have all of these mutations on the top, plus some of the newer ones that are generated more recently. Good, so much for the phylogeny part, and unless you have questions here, I will end this part with making a small comment on the more complex aberrations.
One question?
Yes.
Go back one slide. So, did the authors of the original study do any collapsing of mutations together? So, is this a result of clustering, or these are the individual mutations?
These are individual mutations, and they are just...
They are not representatives of larger sets, so they all...
No.
Because it looks like a miracle that you don't have any two collapsing together in this.
What do you mean by collapsing?
That you can distinguish between them. You always have some sample that can distinguish between any two consecutive nodes in this very long chain.
Yeah, almost. I mean, like the first three, for example, okay, there's this, maybe these two I cannot, okay, I didn't check this in detail, but I'm not sure it's true what you're saying. And of course it depends, I'm just showing you, again, the optimal assignment here. I mean, what we are really interested in is just the tree on the left-hand side, right?
No, but then the tree may have some degrees of freedom.
Yes. I mean, maybe I should say in general, we also look at other data sets, I will not show them today. In general, that does frequently occur, that there is a group of mutations that occurred that we cannot distinguish because we have no sample here, right? In general, that is clearly a feature, and that's what you would expect from very ancestral mutations, that there are just no cells left, or not many at least, that would be able to distinguish. Absolutely.
Possibly related to that, what's represented by the multiple arcs? You know, so like from the fourth purple thing, DMXL1 to S6, has like 6 arcs connecting them?
Yes, so again what I'm showing here is the optimal assignment of samples to the mutations, and often that assignment is not unique. So several assignments have the same optimal probability, optimal likelihood. And then I'm that figure, I'm showing all of them.
Even when the same two things are being connected? So like S19 on the top right is connected to 6 of the genes.
Yes.
But then S6 on the bottom right has, looks like 6 connections to one of the genes.
Yes.
Bottom, fourth row up, you're close to there.
Yes.
Oh, yes.
So they're like
Which one?
S6.
S6.
Right.
Yes.
Yeah. It's like it has multiple connections, like not that one, but the lower.
Yes.
I think it's just the curves of the others mixing up.
Okay.
And the question is why that is. Yeah, the question is again that the assignment is not unique. I mean, really what we're doing is we're integrating this out, and we're only looking at the tree on the left-hand side. But I'm showing you this figure to give you a feeling for the uncertainty that one has in assigning these samples. And in other words, I'm doing this to give you additional reason to integrate this out. It is in a way a nuisance parameter. It would not be possible in a meaningful way to decide for one of these assignments. That's the whole point. Whereas the structure of the mutation tree on the left-hand side, I mean, it's the same on the right-hand side, but the structure only among the red vertices, that one is robust and is meaningful.
So, Niko, maybe the following question will be trivially true, but answerable. But the possible assignments, are they all going to form a connected compound?
I don't think so. I don't know, and I haven't thought about it. That's of course that's in a way the most obvious reason. So, but I'm not sure that this is that one can prove it. Yeah, no.
Can I ask a question?
Yes.
I know the input to your algorithm is like a mutation sample matrix.
samples are linked to the same mutation, are they actually mutated at the same position or completely different positions? Different positions of the gene.
They will, I mean, of course, in a way that's all encoded in the probabilistic model in the likelihood, right? But they will tend, I mean, the idea is, of course, okay, so, I mean, basically it's like this. If you, okay, let's maybe go to the ideal setting here. So the way, let's say this sample 6 down here is defined, is you trace out the path from the root to that sample, and all the mutations you pick up on that path. So in other words, sample 6 would have exactly mutation 2 and mutation 3.
You have the same gene being mutated. I mean, when you go and look at the sequence of the gene, are the mutations at the same location?
Okay, I mean that's a matter of how you define these. What do you mean by a mutation here, right? How you encode the binary matrix, and you can say, well, I want to look at the very specific single nucleotide variant, for example, or you can say, okay, whenever the same gene is mutated in a similar way, then I summarize that as the same mutation. Discrete events, yeah.
Even though at the end of the tree has genes, even though.
I mean, this is only in this specific application where where the mutations here have been summarized per gene, and therefore the nodes here are labeled with genes. But you could apply it in the same manner to, I don't know, SNVs of one gene, for example, or whatever, right? Or other genetic events that you measure.
So can I ask a question that probably I had asked like 10 minutes ago? Why do you imagine to have like same tree or same structure, same order between mutations for all the patients?
This is for one tumor, for one patient only. We're making here inference from one tumor, and I'm only saying this is most likely, according to our model, what has happened in this one tumor, or in the sample we got from that tumor.
Oh. And then you also imagine that you don't have copy number.
Exactly, but let's say it's in a way more abstract. Yeah, I mean, in a way it's more abstract because we're just saying there are these mutational events, and in principle that could also be a certain rearrangement, and you just record is it present or not in any of these cells. So this is not linked to any specific type of mutation, but on the other hand it doesn't account for the dependencies of these mutations. They are not independent in a real tumor, right? And that's, I mean, the whole starting point of a binary matrix is, of course, very simplifying. Okay, more questions at this point? Yes?
So in the next graph.
Yes.
Yeah. So for example, S9 has all the mutations? You would still see the
where's S9?
Like,
here. No, S9 has all of these mutations. Boom. Sorry?
Yeah.
Did you point to another one?
Could you have some of the, for example, S19, you have all the arcs, but
for S19.
You're not sure if some of them are there.
That's the fact that there's multiple arcs to S19 reflects my uncertainty to assign S19 to one of the nodes. I realized that I should remove this figure from my next presentation. I thought it was a nice graph. Okay, did you have a question? No. Okay, let me move on and finish this part by, again, briefly mentioning that things are more complex, and you have already alluded to that, yeah, what about deletions and so on? And yes, life is more complicated. And there are a few early attempts to get at the, let's say, rearrangement tumor phylogeny. Chris Greenman, for example, will talk about this at the workshop here. So in reality, more complex things can happen, as we discussed earlier today. So for example, here's an example where you start with a normal genome, and then you have complex genetic events. Here's an inverted duplication, a breakage fusion bridge cycle, and a chromosomal duplication, and so on. Many other things can happen. So the end result, in general, is not easily to summarize in a simple binary matrix, and S and V's alone, again, will probably not do the trick. So there are efforts in trying to reconstruct the rearrangement tumor phylogeny, but it turns out to be much more complicated, and there's no simple problem here to formulate. Okay, with that, I would then like to... Oh, it's already fairly late. What am I going to do now? I give it back to Ben now. So the plan was in the second part to talk a bit about population dynamics. Where's my chair here? It's you, right, Cenk? What are you saying now?
So let's perhaps have a little summary for 5 minutes, and if you'd like to continue tomorrow.
Okay. We are continuing tomorrow anyways in a session, so I give the 5-minute version of this.
I won't be the chair tomorrow.
Okay. So the point is that all these phylogenetic methods, they of course care about the tree, the genealogies, so they care about estimating the dependencies among the samples or maybe among the mutations, but they have no notion of the dynamics of this process. There's no mutation rate in the first place. In particular, there's no population size. The population size plays a big role, and population genetics models address this aspect. And I had put together a few very simple examples to illustrate that point. The first one is the activation of oncogenes. Now, oncogenes are simply those genes, let's say they are driver genes, but they are those driver genes that can be activated by a single event that's either a point mutation on one of the chromosomes, or a gene amplification, or chromosomal fusion. So that's the way to activate an oncogene. And the dynamics of this process are very simple, because you have this one event. So the probability that such a mutant with an activated oncogene has been fixed in the population of tumor cells by time t simply follows exponential distribution. So it's 1 minus e to the minus, and then the rate here is nu rho t, where n is the population size. So here it matters how large the tumor is, how many cells there are. U is the mutation rate, and rho is the fixation probability. So the probability that once that mutation has been generated, it will actually reach fixation in the population. So the way to think of this is that n times u is sometimes called the total mutation rate. That's the total chance of generating the mutation in the population. And now it's intuitively clear that the population size plays a big role here. In a larger tumor, you have a higher chance of generating that mutation as compared to a smaller tumor. Rho, again, is the probability that it will eventually reach fixation. That can be computed under simple population genetics model, Moran process, and so on, for example. So this very simple model already shows you that large compartments accelerate the accumulation of advantageous mutations, and small compartments, where there's a small number of cells susceptible to such mutation, will slow it down. And in fact, most tissues with a high cell turnover, where cancer can occur and does occur eventually, are organized in many small such compartments. And the typical example are the colonic crypts in the colon. So in a way, you can argue that these tissues are organized in a way to prevent cancer as much as possible. So having a large number of cells organized in small compartments is in a way good protection against the activation of an oncogene. Now, the other class of driver genes are the tumor suppressor genes, and in a way these are genes that protect you from cancer. They are recessive, so in order to inactivate them, you sort of need two events. For example, 2 point mutations, one on the one chromosome and the other one on the other chromosome. Or, and actually that's very common, you have one mutation on one of the chromosomes, and then you have a loss of heterozygosity event. You either lose the second copy or you just lose the heterozygosity, so you just end up with 2 identical copies, and therefore both of these copies are affected. So in other words, in order to inactivate a tumor suppressor gene, you have to affect both copies of the allele. Now, the dynamics of this process turn out to be more complicated, and I'm not showing you how this works, but I'm giving you the basic result here. And the dynamics of this process depend very much on population size. Now, for small population sizes on the left here, the typical scenario looks like this. You start with cells that have none of these events, and then here comes up a subpopulation that has the first hit, but it goes extinct again. This one again has it, goes extinct. This one eventually reaches fixation, and then after it has reached fixation, the second event occurs here. So here the tumor suppressor gene is inactivated. So in other words, this is a 2-hit scenario. You first need the first mutation to reach fixation, and then occurs the second one. Now, for intermediate population sizes, it turns out that again you have the first mutation coming up, but typically the second mutation occurs while the first one is still fluctuating and before it has reached fixation in the population. This is what people sometimes call stochastic tunneling. So you don't need to wait for the first one to reach fixation, but the second one typically occurs much before that. So in terms of the rate-limiting steps, that's a 1-hit process. And finally, in very large population sizes, mathematically it looks like 2 hits, but they are not rate limiting. So in a way here it's immediate, right? You immediately grow with your type 1 mutant, and the second one occurs immediately. So it goes very fast, in other words, in large population sizes. Okay, the third part I wanted to address was the progression of tumors, and I will show you only one slide here. Namely this one. This is a simulation from a model where you start with something like 1 million cells here, and there's a certain evolutionary process that just accounts for mutations accumulating in the tumor. And the different curves here correspond to different clones defined by the number of mutations. So you start with no mutation here, then comes here the clone that has 1 driver mutation, and while this clone is growing, you have here the first cells with 2 mutations. And how do you get a cell with 2 mutations? Well, a second mutation occurs on the growing clone with 1 mutation. And you see that's initially a very stochastic process. They go extinct, they come back, and so on, and then only when they reach a certain size, they grow almost deterministically. And overall, these mutations come almost in the clock-like fashion, right? These selective sweeps go through the population, and in this manner, mutations are accumulated. Okay, I will skip a bit here and just make one more comment in the end, that you can also use these types of models to predict the effect of treatment. Mono versus combination treatment has been mentioned at some point before. Here's such a study using these types of models comparing monotherapy to dual therapy, so treatment with 2 compounds. And the total tumor load is shown here in this greenish color, and the subpopulation of resistant cells is shown in red. And as you see in the top row, depending on the parameters of the model, you have a much harder time reducing the load of the tumor and also preventing the development of drug resistance, which eventually will lead to recurrence of the tumor. Okay, with that I would like to end this part, and on this very last slide I just compiled a few things that might be interesting or even special about cancer development and cancer evolution, because I know there's a number of people here interested in this process from an evolutionary biology point of view. I think these tumors are very interesting evolutionary systems. One aspect we emphasized a lot in the first session today was that cancer genomes harbor very complex alterations. The second one, that in the context of networks, I think became very apparent, is cancer is the evolution of defection, because there's many ways of breaking certain signaling pathways, for example. And that's one reason why there is many selectively advantageous mutations available for cancer to evolve. So there's many drivers, basically, because there's many ways to interfere with these signaling pathways that usually control the cells. And once you interfere with them, these cells go wild and they don't behave in the tissue properly anymore. They replicate more than they should, and so on. Tumors harbor specific population structures, of course, in a way that's true for almost any evolutionary system, but also, let's say, in tumors, at least or especially in solid tumors, that plays a big role. And the fate of mutations depends a lot on the structure. I didn't have time to go into it, but the fixation probability and the fixation time of a mutant depends a lot, not only on the usual parameters like population size, mutation rate, and so on, selective advantage, but also on the structure of the population. And finally, and this points to what I will be talking about tomorrow morning, one special aspect of this evolutionary process is that if you collect data from different tumors of the same type, as we are doing, of course, at a large scale, then you have IID data from these tumors. So there is an evolutionary process that is repeated independently in different individuals. Of course, not exactly the same, but it's a fairly similar process. And that's, again, something very unusual for an evolutionary process, because most of the evolutionary processes, we only observe the end result of it happening once, right? And you hardly ever have a chance to repeat this stochastic process. And with tumors, you have this IID data. So I think this is a big difference, and this gives us a lot of opportunities to modeling this evolutionary process, to look for parallel and even reproducible evolutionary processes. So how much of this process is actually reproducible if it occurs in different patients? And the question then at the end of the day, how much of this evolutionary process can we actually predict? So phylogeny very much takes the point of view into the past, trying to reconstruct the past history. But the data that we have, I think, can do much more, and we can ask how much of this evolutionary process can we predict, and at the end of the day, use that to make predictions that hopefully are clinically important and relevant. Thank you, and I apologize for going over time. I don't know, very quick question maybe, but yes.
So just to relate to the last point here, actually, how do we know that the tumors are from the same type if they are?
Yeah, that's a good question. I mean, of course, the way we do it now is the tumor types are at least mainly categorized by histology, right? And so that's probably what I mean at this point. But I think your point is very well taken. Is that classification really the right one or the best one? So exactly, it comes back to the subtyping problem that we mentioned earlier based on the molecular profiles. But even then, every tumor will be different. That's clear, right? I mean, of course, you can say, no, this doesn't make sense because every tumor is different. And that's probably a fair perspective. But the hope is that some are similar enough such that you can learn common features of those.
All of this was talking about clonal expansion that looks like a tree. I've seen some papers recently, biological papers, that suggest that there can be some reticulate behavior in the expansion of the cells. There's tumor cells in different organs somehow communicate and affect each other. Is that?
I don't know much about it. I don't think there's a lot established. I mean, there's, of course, no recombination here per se, and other, I don't know, lateral gene transfer and so on. I'm not really aware of it. I don't know. There's certainly very, very little, and that would, of course, complicate things massively again. Yeah, I don't know. I think at this point we have no strong evidence for that.
Was it about this breast cancer and liver cancer? The woman have just two cancers.
I read Science and Nature, so it must have been one of those. But I can't say specifically.
There are clinical observations that suggest that.
I see.
Small R